techniques that are used in rDNA technology. So it is used, different techniques are used to obtain the proper information during the genetic engineering process. So techniques like gel electrophoresis, blotting technique, dot blot hybridization, DNA sequences, PCR, it means polymerase chain reaction, various techniques are used. Starting with gel electrophoresis, so it involves the separation of charged molecules under the effect of electrical field. So molecules are moving on the gel towards oppositely charged electrode. For example, cation move towards the negative electrode. Negative electrode, we can say it is anode, and anion can move towards the positive electrode, we can say cathode. So here the molecule is moving towards the electrode. Now these molecules are moving on the gel. That's why it is known as a gel electrophoresis method. Now the genomic DNA is obtained from the desired host and fragment into the plasmid, or we can say restriction site of the host cell. Now the gel electrophoresis utilize the separate out the fragment and isolate the desired DNA fragment. So by this technique we are separating the cutted fragments of DNA and isolating that fragments. Agarose gel is commonly used in gel electrophoresis, and for the large segment of the DNA the agarose gel is used, and for the low DNA, or we can say small DNA fragment, polyacrylamide is used as a gel. Now in this process buffer system is used for a gel medium and apply the source of direct current. Now next in this the DNA fragment moves up to the different distance on the gel on the basis of charge to mass ratio. If the DNA fragment having the heavier, so it cannot travel to a larger distance, and the lighter DNA fragment travel too far, or it travel to the more distance. Next is blotting technique. So it is the transfer of the macromolecules from the gel to the surface of immobilized membrane. In this technique, ultimately the immobilized membrane, example are nitrocellulose membrane, nylon membrane, carboxymethyl membrane, these type of membrane are used, and on this membrane the macromolecule is transferred. That macromolecules are present on the gel. So from gel it transferred onto the immobilized membrane. Now blot transfer technique having three basic type: southern blotting, western blotting, and northern blotting. So southern blotting is named after the scientist E.M. Southern, who developed this technique in 1975. So these three techniques are divided based on the separation mechanism. So this southern blotting is used to transfer the DNA from gel onto the membrane. Northern used to transfer the RNA, and western used to transfer the protein on the membrane. Now third, or we can say the fourth method is southwestern blotting. So this blotting technique is examined the protein DNA interaction. Now next is dot blot hybridization. So it is the same as a blotting technique, but here difference is that the DNA fragment separated by direct applying them onto the dot on the nitrocellulose membrane. So here this method is same as blotting technique, but here the small difference is that the DNA fragment applied on the membrane, and on the membrane it is applying as a dot. So here is we are not using electrophoresis process.